right. Hello, everyone. Welcome back. Feels really weird to be on campus again. Hopefully, our brief interval online wasn't too disruptive for everyone. Um, I was in the process of typing up an announcement this morning, but I wasn't quite sure how long it would take to get in this morning. So I left a little bit early. So I will be posting an announcement very similar to this on Moodle this afternoon. But I basically just wanted to say that you might have already noticed that you have access to your midterm one grades. I've also given you access to all of the questions and the answers and any feedback as well. Usually I do like in-person midterm reviews, but it's really difficult to do nowadays, especially where sessional instructors don't have offices. So I just figured I'd give you access to everything, and then that way you can go through and review the questions, review the correct answers, and then if you have any specific questions, you can take a picture and email it to me, and we can go over it that way. Or if you want to go over a bunch of them, we can set up a meeting. That's fine too. Um, I have mentioned to a couple of students that I've taught enough sections of intro that I have a particular distribution with the grades that I know and what to expect. And anytime I have any deviation from that, I'll usually try and make some corrections. So if I have the grades are just a little bit too low, then I'm going to do some adjustments and bring it back into line with where they should be. So what I found with this exam was that students either did really, really well or they didn't do as well. So I had some people who did fantastic and other people who it just didn't work for them to be doing the exam online. And because my whole goal with all of my different kinds of assessments is to try and get like a um, representative score, I guess, of your abilities, having something like this outside of our control where we had to swap our method of delivery over, it's really not that representative. So I'm making two adjustments that are going to help boost grades depending on how things work. Whenever I'm making these adjustments, I will never drop anything. If you guys do better than what I anticipate, then fantastic. Great job. If you don't do as well as I anticipate, then I take that as a me problem. So that's why I'm making these two changes. The first is that I am adjusting what the score for the midterm is out of. I didn't have any one question that everyone did poorly on. Usually I just drop a question. But it was everybody had different issues in different places. So what I'm just going to do is score that's in Moodle is treated as out of 45. I'm going to treat it as out of a smaller denominator. So if you have, I used the math here of 30 out of 45, that was a 67. I can't remember what the exact number is. Like I said, I wasn't on my desktop this morning. It's going to be even a little bit better. I think I'm doing a smaller number than 43, but it's at least 43. So I'll be dropping the equivalent of two questions or more. But I'll give you those exact numbers later on. So when you're calculating that out, once you see this announcement come out, use that new number to calculate your average for the test because that's what I'll be doing when I put our grades. So hopefully that one makes sense. For the second thing I'm doing, for those who ended up really struggling with an online delivery method, that is beyond our control. That wasn't something we could control. So what I'm going to do is I'm going to have a second grading scheme. Basically, I'm going to calculate your grade two ways. One is the standard one that was in our syllabus. So as is, straight from our syllabus, where the midterms are both worth 25%, the finals 30, no changes there. But I'm also going to calculate a reduced weight for this first midterm. So I will check what your grade would be if we gave the first midterm only 15% of the weight instead of 25. And then I'm going to move that extra 10% with the second midterm and the final. All of the details are in the announcement that will be coming out. But just basically, if that gives you a better mark, if it does better for you to be using our in-person exam scores versus an online exam score, then I'm going to bias it towards the in-person one. If you're better off, if you did great on the first midterm and that's going to do well for your grade, we'll stick with that one. I'm just going to calculate both and take better. So hopefully these changes here can help alleviate a little bit of concern because I know that online tests aren't everybody's thing. And our in-person tests are not as hyped. So hopefully from here on in, we can go back to our standard appearance of tests. But I'm doing this just so that we can bring it back into sort of where we should be in terms of grades. Yep, a little bit, yes. So I set it up. With online tests, there's a couple of ways you can go. You can either use one of the proctoring softwares, which I have seen go terribly wrong, and I do not enjoy that. So I don't use those. What I do instead is I make it a little bit tighter on time, and everyone's exam is unique. So I built an exam that was probably 3 times the size it needed to be and set it up to pull randomly from a larger data set. So I divided them up by topic and by chapter and then pulled from those. So everyone got a unique test, which is also why it's really hard to do things like exam viewings without just giving you access. So yeah, when we do in person, it'll be more just on paper, probably 1 or 2 versions, and like that's it. So, and especially with the in-person stuff, I will again bias it towards less intensive questions. With online, I'm just worried about like people can go on Google and just look up answers. So that's why we're tight on time online. But in person, that's not really a problem. You have your notes, you can make a cheat sheet, you can work that way. So I'd probably drop the total number of questions by probably about 5. So yeah, that's the way that works out. And I like to be transparent about why that stuff happens the way it does. So hopefully all those extra details, despite being more than you guys probably need, just helps them. Cool. All right. So I will be putting this out on Moodle as soon as I get the exact numbers, which are on a different computer. But just so you know that that's coming, I figured if we were going to talk in person, we do it that way. Um, yeah. Uh, Scroll back up to not the top. There we go. Okay, I think that was everything I wanted to cover about the midterm. The next thing I wanted to mention, as you can see, I have my whole setup here again. So if you find it annoying to go through the security check or whatever on your way in, if you're not feeling like coming in, I'm still going to be putting everything online. So you don't have to be here if you don't want to. Um, we are going to do sort of our middle portion of chapter 4, which I keep calling chapter 5 and catching myself. In every other class I've done, this is chapter 5. Same topic, just in a different place. So it's been throwing me for a little bit of a loop. But as you have noticed, and I think I discussed it also in the recordings, I do this chapter a little bit out of order. And that's because every textbook I've seen decides to launch right into vision. And vision is one of those really big, complex topics, and they start off right at the top. And I'd much rather sort of work our way up to vision, develop that understanding of the terminology and the concepts to sort of work through, say, our auditory system, which is very similar in a lot of ways to kind of get us into that mindset. And then we conclude with vision. So a lot of this stuff is a little bit out of order, but I find that this order makes more sense to me at least. So if you're reading the textbook, it's the same stuff, just in a different order. And so today we're going to talk at first about our chemical senses, so olfactory and gustatory systems. And we're going to talk about sort of our bodily senses, things like touch, kinesthetic, and vestibular systems. And you're going to notice right off the bat that in comparison to what we already saw in our auditory system, these are going to be very brief. These are just sort of little bite-sized introductions to these topics. And there's a couple of reasons for that. One being that we don't have a lot of time to go into tons of detail on each of these. But the bigger one, and the one that I like to talk about, is the fact that with a lot of these systems, we don't actually know the specifics of how they work. You'll find as I'm explaining some of these things, I talk about sort of research studies and things that some people have shown, or I might talk about things that this is how many we can agree on, but not everyone is in the same boat. And that's because a lot of these topics are just being sort of revolutionized and changed over. So as our technology has improved, as we're getting down to, say, single neuron recording, it's getting a lot easier to understand how some of these system works, but a lot of what we know is still theoretical. So we don't go into too much detail for that reason as well. All right, so let's start with our sense of smell or olfaction. And as per usual, I'm going to talk a little bit about what is our physical stimulus, what's causing this sensation, and then also what our receptors are and how the system works in a lot less detail for these guys, though. So, our physical stimuli for smell are odorants, or even just chemical substances that are carried in the air. My favorite description that I've seen from a textbook was that they are volatile chemical substances, which, from the scientific perspective, is accurate. Volatile just means that they evaporate at room temperature. And chemicals, because everything is chemical. But it just sounds so intense the way that they had worded it. But what that just means is that most of the things that we smell are things that evaporate at room temperature and are basically dissolved in our air so that we can bring it into our noses and it can then dissolve in the fluid, the mucus that lines our nose cavities. So our olfactory receptors in this case are called olfactory cilia. And all of the images for this, every textbook has some iteration of the exact same image. And I find it all very strange and unhelpful. But if you've ever had a sinus infection or had that experience sort of at the back of your nose, above your mouth, where you get that burning feeling, that's your nasal passage, that's your sinuses. So we have lots of open pockets in our heads below our brain. And so some of those are connected to our nose and have a mucus lining where stuff can dissolve. And so once those chemicals have dissolved in that mucus, they can be picked up or bind to our olfactory cilia. These are our receptor cells. And they're going to move that signal along those receptors up into our olfactory bulb, which is then going to relay them into the brain. So we have a full pathway here where we also include transduction. I've realized that I didn't include the stage of transduction in all of my pathways, but it always does happen. So we have those chemicals bind to our olfactory cilia, they bind to our receptors, and it transduce into a neural impulse. So they're firing the action potentials in our neurons. And here that's going to transmit along our olfactory nerve and into the olfactory bulb at the base of our brain. Now, our sense of smell is unique in that it doesn't route through our thalamus like our other senses tend to. When we talked about the different parts of the brain and the role that they play, our thalamus was that routing station that sent stuff to the parts of the brain it needed to go to. But our sense of smell doesn't, because that olfactory bulb touches right on the base of our brain and can distribute into the brain on its own. So it's the only of the senses that we're going to talk about that doesn't involve the thalamus. And the reason for this, at least theoretically, is that we evolved a sense of smell very far back in evolutionary time. So this is a very basal skill or a basal sense, meaning that we shared a common ancestor with lots of species a long, long time ago that could smell, probably before our brains even had a structure as complex as a thalamus. So the idea here is that this is one of those early evolved senses that we've kept around and hasn't modified a ton. So it doesn't go through the thalamus like everything else does. It's kind of neat as someone who took some evolutionary biology. Um, so our sense of smell ends up showing sensory adaptation, pretty much like all of the senses that we're going to talk about and have talked about. They all show some kind of sensory adaptation. And I love using the example of the Febreze nose blind commercials because that is exactly what's happening. So the perceived strength of an odor will fade, and so you lose about half your perception of strength within as little as 4 minutes. So in the Febreze commercials, they show that you get in your car and it's fine, you don't notice anything. You go pick somebody up and they get in and it smells like dog. And that's because you've gone nose blind. You've been in the car for long enough that you don't mind the smell, you don't notice it anymore. You've adapted to that smell. But to other people who aren't used to the smell, it's a lot more strong, it's a lot more overpowering. So we experience this all the time. You walk into a bakery and at first it smells really, really good and you're really, really hungry. But if you have to wait in line for a long time, you stop noticing the smell. Instead, drop off as you adapt to it. There's also some really cool research looking at our ability to perceive smells. And so there's a lot of research that shows that females tend to be more accurate than males in odor recognition tasks. So they're a lot better at categorizing what smell they smell. Um, but I also like to point out that humans, we are very not sensitive to smell overall. If you look at our capacity compared to something like dogs or most other mammals, we're very bad at smelling. We need much, much higher concentrations of an odor for us to be able to perceive it. For other species, it's a lot easier at lower concentrations. I think, if I'm remembering correctly, something like bloodhounds, their sense of smell is like 40,000 times stronger than ours. So they can smell just a couple of the chemicals dissolved in their mucus, whereas we need thousands of those same particles to get any kind of reaction, which is really neat. Um, I do also point out that we have some variation here where some people have a better sense of smell than others. Some people are really good at telling smells apart, some are not. I go against all the research and I have almost no sense of smell and have never had a sense of smell, which has caused some problems. But that's just since I was a kid, I have like four scents that I can tell apart, and everything else is it smells strongly or nothing at all, which is interesting when people don't realize that about you. It's also been really fun with COVID symptoms for people because I don't have a smell and I never have, but that's one of those things people watch out for. So just a lot of fun to explain that one. But our sense of smell is tied in very, very closely with our sense of taste or sensation. And so here we have the same basic idea. Our physical stimulus are those soluble chemical substances, things that are going to dissolve in our saliva. So the same way that our smells dissolve in the mucus in our noses, with taste, we have saliva that dissolves tastes in our mouths. And these receptor cells are found in taste buds. Though I do like to point out to people that what we think of as the taste buds, those bumps that are on our tongues, those are not actually taste buds. The taste buds are in the grooves between the bumps. I do have an image here. So if you look, the taste buds are in those little valleys between the bumps. So when you think of tasting something, it's not actually the bumps themselves that are doing the tasting, it's the spaces between them, which I thought was really, really interesting. And we have different kinds of receptors. This is another one of those. We have 4 or 5 or more receptors. Most texts will say that we have four: sweet, salty, sour, and bitter. More often now than not, we start seeing mention of a fifth flavor or a fifth taste, which is umami, which is sort of a savory taste. And I'm including it because I'm seeing it more and more included in the literature, and there's a lot of studies that are coming out that include it as well. But most of the images that we see will only refer to 5 or 4 instead of 5, just because the fifth one is newish. And I do like to make people aware of that tongue taste map you're shown usually in grade school is very inaccurate. You usually get that map. It's like, oh, you can taste bitter at the back and sweet at the front, and then sour and salty on the sides. But that gives you this false sense that you can only taste bitter at the back and you can only taste sweet at the front. That's not actually the case. So if I hop back to this weird map of the tongue, we actually have those graphs at the top that show the distribution of those different receptors. Again, they only have 4 because it's an old image. But what we see here is that yes, the back of the tongue has a high bar for those blue bitter receptors. So there's a lot of bitter receptors there, but there are still receptors for sour, sweet, and salty. So it's not that you can only taste bitter there, it's just that we have a higher concentration of bitter at the back than in other places. But if we look at our other graphs, sort of the middle and the side of the tongue, we still have bitter receptors. And we have stronger sour receptors. So we have different concentrations of these receptors all across the tongue. And this just looks at three different locations. So the front shows salty, uh, both of the back ones show bitter as the highest concentrations. Different places are going to have different distributions. Yep. Um, that one, I don't know they've even done that yet. Because I haven't read too much on the new literature for it. Even like these taste maps are fairly recent, like in the last 5 years or so, I think. Because it used to be like, yep, there's you can consider it back in that day. So I would assume that it's fairly equally distributed, but I don't know where would have the highest concentration. Might be something interesting. Yeah, I'd love to get an updated version of this graphic because this one's been kicking around for a couple of years. Oh, yep. Ooh, there might be a couple of different ways you could do it. I don't know the literature super well off the top of my head, but I would think that either they do very controlled, like titrations of substances that we know are sweet, salty, sour, bitter, umami, and place it on specific places and ask them, like, how strong is that or how not strong is that? And just see if they can, like, get a good measure of strength based on concentrations. Another way they might be able to do it, I don't know if the individual receptor cells look different. So they might have a different appearance. So if you could do like a dissection of dead tissue, then you might be able to tell that way. But those are the two big approaches that I'd probably think of. I think what else. Oh, there's also one. I don't know if this is from an actual science show or if it was in a movie, but they can put a sensor on the tongue that has like electrical stimuli, and they can stimulate different parts of the tongue, and that will correspond to different tastes. So that might be another way they could do it without having to get too invasive. Yeah, yeah. So that's those are the like, the big ways that I would think of to approach the question. Cool. Um, all right, and then this one here, I just wanted to basically show you that each of the taste buds, so the clusters that we're looking at, sort of in those, you can see at the very top that little bit of a groove here. There it is. So that would be that valley in between the bumps that we used to think of as taste buds. We have this cluster of our receptors in that valley. And they're going to contain somewhere between 50 and 100 taste sensitive cells. And they're going to be different kinds of receptors for different kinds of flavors or different kinds of tastes. And it's the same basic layout. What I want us to get out of this here is that we have the receptor. They feed back into an efferent here or here, an efferent nerve. So we're sending that signal out and towards the brain. So the same basic setup as everywhere else. And so for our pathway, we have the taste buds that then we have that transduction into a neural impulse, and it's going to route up through our uh, into our brain at the thalamus, and then the thalamus is going to distribute that information to the rest of our cortex. So we can then do with that as we will. And we will talk very, very briefly in just a moment about how taste and smell combine. But first, I wanted to talk about basic taste preferences. So there's some evidence that some of our basic taste preferences are innate, meaning that they might be biologically encoded. So if you think of pretty much every child you have ever met loves sweet things, and that might be for a reason. So if we think about it, a lot of things that are sweet are also high in calories. So even if we don't look at processed sugars, even if we just look at naturally occurring things that are sweet, things like berries, those tend to have lots of energy, lots of nutrients that might be beneficial. So if we consider humans not in, say, today's society, but living in nature, When you're younger, you need more energy, you need more resources to continue growing. So it makes sense that they would seek out things that tend to be calorie dense, which help them grow and develop better. And it's really neat because there's a lot of evidence that our preference for sweet things tends to decrease with age. So as you get older, it might get a little bit easier to eat things like dark chocolate if you were younger and you couldn't stand it. I know that was a problem for me until my mid 20s or so. But we have that sort of adjustment in our preference for tastes, and we get a more diversified palate as we get older. So there's a little bit of a physiological component there, but then there's also a learned component as well. So what you're exposed to will determine what you enjoy. And with food, especially, there's a really big association between who you tend to eat with. So maybe you have a special meal that your family used to always cook for a particular holiday. So you like that meal not necessarily because it's your favorite food, but because of what it reminds you of. So stuff like that can become very closely entwined, which is very, very interesting. And there's also what you were or were not exposed to when you were younger. So there's a lot of differences between cultures, and it's not necessarily because of any biological factors, it's just what you've been exposed to as you grew up. What is a traditional meal for your family? That might be something that then it's your go-to comfort. I grew up in a Nova Scotian family, so they did a lot of boiled things, lots of fish, lots of potatoes. Didn't have rice until I was like in high school, so very limited exposure, and that means that I'm fine with having boiled potatoes and bologna for multiple nights in a row, even though it's boring because it's what I'm used to. So lots of learned preferences there. And then if we want to combine our taste and our smell, these two signals are going to come together in our orbital frontal cortex, this part of our brain. And we're going to look at flavor, that's the specific term, for this combination of taste and smell, as well as a little bit of that tactile sensation of food. So if you think of an apple, it has that crunchy texture, or granola is more dry. That's also a very important part of the flavor. So when we think of flavor, we tend to kind of confuse what is taste and what is smell and what is whatever. But if you think of a time when you had a head cold, you were really stuffed up, your nose was all blocked up, and you'd have a meal that would usually be very flavorful, but it tasted bland. And that's not because there was less taste there, but it's because you were missing that sense of smell, because those chemicals couldn't get to the receptors in your nose. So when we lose our sense of smell, we lose a lot of our sense of taste, which now you can see why that poses a problem for someone with no sense of smell. I don't have a fantastic sense of taste for this very reason. And we do have, again, within individuals, there's lots of variation in our ability to smell already, but also with taste. So there's a spectrum. So in the middle, we have our normal taste ability. If you have poor sense of taste, you might be classified as a non-taster. It just means that you're very low on the sensitivity of being able to tell what you're eating. Or you can be a super taster, which would be a high on the scale, where you're really good at telling apart different flavors. You might have a friend who can have a meal and pick out each and every spice. That would be a super taster. Yep. Yeah, there's some really interesting stuff. Again, well beyond the scope of the course, but they have like the little tablets that you can taste that make everything taste sour or make sour taste sweet or whatever. I don't actually know how those work. Somehow, because I'm like, okay, so if we think of receptors, kind of like the way we've been looking at our neurotransmitters, and different flavors would activate different places. So it must somehow be interfering with the receptors so that they bind not what they should and they bind something else instead. But I don't know how that would work. It might also be something to do with like the signal itself. So if the way that those are connecting in the brain is adjusted at all, that might affect like senses of taste. I don't know though. It's interesting. Oh, interesting. Yeah, I am now very curious about that. I find every year I learn like new bits and pieces to look into, but I might add that to my list. Um, yeah, so I think that was everything I wanted to say about taste and smell. As promised, these are fairly brief, and just you wait till we get into our kinesthetic and vestibular systems, because I have three total slides in those. But before we get to that, we're going to briefly talk about our sense of touch. This is a little bit more dense. Um, but with our sense of touch, we're basically relying on our skin. So our skin has lots and lots of receptors. This is again another one of those where the texts will agree that human skin has at least 6 types of receptors. I don't know what the upper end of that spectrum is, but there's at least 6 they can agree on. And we're only going to talk about 4 of those. So we have tactile sensors, so feeling things in your skin. We have temperature sensors, one for warm and one for cold. Those are basically going to pick up on how much energy is around. So when things are warmer, there's more energy there. There's extra vibration because there's more heat. Whereas when they're cold, there's less energy, losing that energy there. So we have hot and cold temperature sensors. Then we have pain sensors. And we'll talk a little bit about pain and our perception of pain and some of the weird stuff that our brain can do with pain a little bit later on. But for all of our different kinds of receptors in our skin are up to 6 times, or at least 6 times, I guess. All of these stimuli are going to be some kind of mechanical, thermal, or chemical energy that's interacting with our skin. So if we think of something mechanical, that is like pushing on your skin, physically touching it. We can have thermal, that's the hot versus cold. We can have chemical sensations. If you've ever used like an icy hot back rub and all of a sudden it feels really cold, that's due to chemicals and some other stuff. There's lots of different sensors. And as I said, we're going to just stick to the big ones that we can agree on and actually know how they work. But for our pathways here, all of them are going to operate in about the same pathway. And this is where I realized as I was walking through these slides this morning, forgot to include the fact that there's transduction here. We still have to go from our receptors to some kind of mental signal. So after our sensory receptors, that would be where transduction happens. Everything else is fine. I just left out transduction for this one thing. But once we go from the receptors, it's transduced, it's passed into our nerves, it's going to enter into our spinal column, head up into our brainstem, and then there's a crossover point. So when we were talking about the brain and specifically our somatosensory cortex, when we were looking at which part of the brain deals with which part of the body, there's a crossover. So the right side of our brain controls the left side of our body, and the left side of our brain controls the right side of our body. So that crossover point happens sort of after we've passed through the brainstem. We don't care too much about the specifics there, just that it does happen. And then we go through the thalamus, which then directs everything to the correct place in our somatosensory cortex in that sort of band across the top of our skull in the parietal lobe. So we're going to just sort of keep that basic pathway in mind, but it's mostly recycling terms that we've already talked about, which is kind of nice. So let's talk about a couple of those different types of sensors. We're mostly going to focus on pressure and pain because I've already kind of did the temperature thing, but we'll mention it briefly again in just a slide here. But for pressure, we have specific cells that's in our skin that are sort of connected to particular patches of skin. So our brain knows that a signal from this particular place is from this particular pathway. So we have almost like a little target all across all of our skin. And it works where at the center of that target, at the bullseye point, right where the touch is originating, we have an excitatory center. So we would have increased neural firing at the specific point of contact. And then in the areas around where that contact has happened, because if you say poke on your skin, it's not affecting just the skin cells directly under the finger. The ones that are around it are going to move a little bit as well. And we can differentiate between that direct pressure and indirect pressure. And the indirect pressure ends up getting an inhibitory sort of experience. So instead of increased firing, we see decreased firing around that target center. So that way we get to localize exactly where our skin has been contacted, and our brain can interpret those signals and know exactly what part of our body is being touched. We have differing sensitivity in different parts of our body. So if you still remember about the somatosensory cortex, things like our hands, which are really, really sensitive, have a much larger part of our brain devoted to them. And that's because there are more incoming signals that they can differentiate between. So smaller patches of skin would each have their own pathway to report back to their brain. But other parts of our body might have larger patches of skin because it's less important that they get accurate information. I don't know if everyone had this when they were a child, but you'd play that game where you'd start at your wrist and draw little circles and try and find the exact crook of your elbow. And there's a stretch about 10, 15 centimeters in here where it's really, really hard to tell exactly where you're being touched. If you've never done this, I would recommend trying it. Have someone sort of trace little circles on your arm, move up your arm and try and find the exact crook of your elbow. And you'll find that you can miss by up to 10 centimeters and be sure that you're right. And that's just because they have much larger sensors or much larger areas and therefore are less sensitive. I found it was kind of neat to finally, after all of these years, know why that was a thing. Um, and again, as we've said about other types, our sort of sense of pressure, our touch, is subject to sensory adaptation. So that's in the morning, you put on a new sweater, it's a little bit itchy, and you're not sure if you're going to be able to deal with it. But by the time you're ready to leave the house, you're fine. It's all right. You're never going to think about it again until you're in the stress conditions that you class. Now you're uncomfortably aware of how your clothes are sitting. But that's sensory adaptation, where we tend to ignore things that happen constantly. We only pay attention to things that change. So if our clothes are the same all day, we don't really have to be aware of it. We don't want to waste resources on that because why bother? They're not changing, it doesn't matter. Um, right. And then with temperature and pain, both of these sensors, well, I guess all three, because we said temperature has hot and cold, they're all derived from free nerve endings in the skin. And what this means is they don't have actual receptors, they don't have specialized cells that can convert a signal into something else. They have free nerve endings. So the neurons have their dendrites just sort of out in the skin. And when they receive stimulation, whether that's sort of the hot or cold, whatever they're specialized to pick up on, they're going to then transmit a signal down to the rest of that pathway. With pain, we actually have two distinct kinds of pathways, which is really, really neat. So still free nerve endings are our receptors. We don't have specialized pain receptors. But now we have a fast and a slow pathway. So our fast pathway is actually, I wrote it down so I wouldn't mix it up here. So our fast pathway are myelinated A delta fibers. So if I scroll to the next page, I actually have a visual here. So the red are the A delta fast pathway. And then the blue here are the C fiber. Those are unmyelinated neurons, and they're a lot slower. And so what happens here is our A delta is because they're myelinated, they're traveling a lot faster. And they're also going straight through and sort of not bouncing around in the brain to go to other areas. It's going straight to our somatosensory. The slow pathway, because they're unmyelinated, that signal travels a lot slower. We can't jump between the nodes of Ranvier. And once it gets into the brain here, it doesn't necessarily get immediately aligned to the somatosensory cortex. It might be diffused to other places to be used. We don't care too much about the specifics of that. What we do care about is that that fast pathway, it's fast because of that myelinated signal. It's going to happen within a fraction of a second, and it's going to give us an exact location that's feeling pain. Whereas our slow pathway is going to be slower, more generalized. That's that sort of aching feeling. Instead of a precise individual point that's causing pain, that's an overall pain. I love using the example of, when you kick the end of your coffee table, you kick the leg on your coffee table, and you have that immediate sharp pain, and you have just long enough to go, oh no, it's going to hurt. And then you get that dull ache as your whole foot hurts. That's the difference between our fast and our slow pathways. So one is immediate, exactly where you're feeling pain, and then the other is going to be a more broad, generalized sensation. And of course, we could spend way more time talking about these, but we will continue onward. So if we're going to talk about feeling pain, we definitely want to talk about how our perception, how we can frame pain and how that changes our interpretation of it. We also want to talk about some of the theories of how we do or don't feel pain. Those are going to be our last couple of slides for this topic. But for feeling pain, our perception of pain is going to be influenced by lots of different psychological factors. So our sensation, that the two different pathways is always going to be about the same, but how we interpret it, our perception of it can change, which is why we talk about it in a psychology class. So our expectations, our individual personality, our current mood can all affect our perception of pain. And there are tons of very interesting research topics that we can talk about here. I'm just going to pick some of the highlights, and I want to get the idea across that that whole idea where humans are very individual and we all have different experiences and we're going to interpret things differently absolutely applies to pain. So looking at pleasant photos or being around someone you like can reduce your perception of pain. So back to our toe stubbing incident, if you immediately pull up pictures of cute cats on your phone, maybe you're not going to mind it quite as much. Um, we have a difference in our perception of pain based on the intention of the person who caused that pain. So if somebody intentionally punches you, then you perceive it as being more painful than maybe if they accidentally hit you. And there's lots of different studies looking at sort of different directedness of that pain. Yeah, probably, definitely. There's some interesting, I don't actually even have it on the slide here, but like when I talked about like internal mood, when you are depressed, when you're in a lower state, pain is automatically going to feel worse because you're already feeling bad. That's one of the really hard parts about depression, where it sort of compounds, and the more depressed you are, the worse it feels, which makes you more depressed, which is why it's one of those sort of vicious cycles. Um, we can also bring it back to our placebo effect, where if you give someone a sugar pill and tell them that it's a painkiller, they will perceive a reduction in their pain, even if it doesn't do anything at all. So that's that expectation side of things. And there is a massive amount of research on cultural differences in pain perception. And this is one of those that you kind of want to read with sort of a critical mindset, we can say, because a lot of the research, especially older research, was looking at, is it genetically founded? Is it based on like different parts of the world? What's driving this? But the consensus nowadays is that a lot of this research is based on how we frame things. So in psychology, they talk a lot about individualistic versus collectivistic culture. So cultures like North America that focus on the individual and you care about yourself over others, versus other cultures. Sort of one of the big ones would be in Japan, I think is one of the biggest, where you care about your community. And so the way that you frame something can dramatically depend on your perspective on things like that. So the pain of loneliness. If you're from a culture, if you're used to being alone, it's not so bad. Like maybe going through the pandemic and being stuck alone or just with a couple of roommates was really hard on you, but it wasn't shaking your perception of self. But if you're from somewhere where community is everything and you didn't have a community, it could cause a lot of physical distress. So there's going to be a ton of research on stuff like that coming out in the next few years. I definitely sign up for as many surveys as I can because I really like collecting data and seeing what they come out of it. So interesting in the next few years to see how this chapter changes. But yeah, we could spend all day talking about stuff that influences perception of pain. And when we get into the chapter on consciousness, we'll even talk about how things like meditation and focusing your attention can completely reduce pain or even take it away entirely, which is very, very cool. So for our methods of blocking pain or not feeling pain, we can talk about gate control theory. And this one is described in a very abstract way. And I'm going to say, I know that. We're going to keep it abstract because it's an old theory and it's a little out there. But the basic idea is that our incoming pain sensation, everything has to route through our spinal cord. And so in the spinal cord, there is a theoretical gate that determines if those signals pass through or not. It's not a physical gate, it is a conceptual gate. So the idea is that it's maybe like a neural pathway, something that would inhibit the transmission of signals, maybe. But what we care about is that it's a theory that since everything routes through our spinal cord, there might be something in our spinal cord that prevents some of those signals from passing through. So theoretical gates. There's some evidence for this, but they haven't actually found any kind of neural patterns that match this theory. So there are some other, maybe better explanations that we could go with. And again, we don't know the exact right answer, which is why we talk about a couple of them. Something important to keep in mind when talking about feeling pain are endorphins. So these are the body's natural painkillers. They tend to behave in the same way as something like morphine, where it prevents us from feeling pain. And we have endorphins spread out pretty much everywhere in our central nervous system. And the way that they behave seems to correlate with sort of higher concentrations of endorphins would mean we don't experience as much pain. And again, they keep it vague because it's a complex pattern and we're not quite sure how it works. But what we do care about is that the endorphins seem to be involved in a descending neural pathway that ends up mediating our suppression of pain. So our endorphins are sort of like a go signal that tells this pathway, this neural pathway, to not allow signals to pass through. So likely initiated by endorphins, because again, we're not positive, but it inhibits activity in the neurons that would transmit incoming pain signals. So they're not quite sure sort of how and where and why, but this might prevent us from experiencing extreme amounts of pain. And this makes sense because feeling pain in certain levels can be adaptive. If you have a headache and it's because you haven't drank water, then a headache is telling you to go drink water, something you can deal with. But if you're fighting a bear and you get bit, then feeling excruciating amounts of pain is not helping you fight off a bear. So these pathways would allow you to be aware that you are injured, but you have other things to deal with right now. So we're not going to pay attention to those. So the sensation is still there, but our perception of it changes dramatically. So a very, very cool topic that we could talk endlessly about. But I would like to finish our last three slides where we talk about the kinesthetic and vestibular system. And as promised, these are really short. So our kinesthetic system monitors the position of the parts of our body, basically receptors in our joints and muscles that tell us how much our limbs are bent, if our muscles are taut or not, and just basically tells us what our body is doing. Are we standing, sitting? Are we curled up, spread out? What's going on? And that's going to follow the same pathway as our tactile stimulation, so through the spine and into the brain. Our vestibular system is actually something we've hinted at already. That's because it's tied into our auditory system. So remember those semicircular canals, those tubes in our inner ear? Those are fluid-filled tubes that interact with gravity, and we can interpret that sort of movement of fluid to tell us what our orientation is in space. So if we are upright, then gravity is pulling straight down, we feel fluid at the bottom. If we turn our head quickly, we would feel a sloshing in that direction. So if you've ever sat on a office chair and spun around really fast and stopped suddenly, and you feel really dizzy, it's because your visual system is saying that you're stationary, but those semicircular canals, the fluid is still moving as though you're spinning. And that contrast, conflicting information, makes you dizzy. So if you've ever had an inner ear infection and you felt dizzy and had trouble with balance, because it's messing with that fluid in your ears. So this is the biggest point of this here. And what I always tell people is I will usually ask a question about the difference between our vestibular and kinesthetic system, because we only talk about them very briefly and they're mentioned sort of as a pair. One is more on orientation, so how is gravity affecting you? And one is on physical position of the rest of your body. So it might be a good idea to be able to tell them apart. But I will leave us there for today and we'll wrap up on Wednesday with our visual system.